Right, so this week we're going to leave bacteria for a while. And we're going to think about viruses. Okay, and even before I start out with the plan and tell you what we're going to be looking at, one thing you really should remember is that viruses are everywhere. Any kind of living organism has got viruses that infect it. Okay, so most of the time you're going to be worried about, I know, in human biology, viruses that cause human disease. Okay, like rabies virus. If you're interested in agronomy, plant biology, plant viruses have a significant effect on crop yields for many different types of plant. So they're very economically very important. But of course, you know, it's not just higher plants and animals that have viruses. Even the simplest organisms have got viruses. Bacteria can be infected by viruses, which are called bacteriophage. And then everything in between also gets infected by viruses. So all kind of simple plant life, all kind of invertebrates, they all have viruses that infect them. So really viruses are a very important part of the living world. Most of the time not doing anything that's useful for the host organism. They're just parasites. For the rest of the time, okay, we're not going to be concerned about viruses that infect plants, that infect insects, and this kind of stuff. We're really, today and on Thursday, just going to look at a couple of cases. Viruses that infect bacteria, because that fits in quite well with the rest of what we've been doing in bacteriology. And we'll also look at pathology of virus infections in humans. Okay, so what we're going to be doing, right? First thing, what are viruses? Okay, and what makes them distinct from bacteria and all other forms of cellular life? And a little bit about how we can study viruses. How do we know anything about these biological objects? And then some of the basics about virus structure and virus replication. So this is a kind of general introduction. So this will be true for all viruses, including viruses that infect bacteria, bacteriophages. And then we're going to carry on Thursday. Maybe we might start a little bit with this. I'm going to look firstly at viruses that infect bacteria. How do they replicate? We'll look at the lytic and lysogenic replication cycles. This will be very important to understand. And then we'll have a little look at virus infections in humans. And probably I'm not going to do smallpox this year, but I'll be covering Ebola virus hemorrhagic fever, because it's a little bit more topical. Well, last year it was, but this year perhaps we can celebrate the fact that it's not infecting people anymore. Right, so what is a virus? Now, the first viruses, actually, if we go back to this slide, you know, the first viruses were really studied perhaps in the 19th century, end of the 19th century by Pasteur, and then by other biologists a little bit later on, the tobacco mosaic virus. So for Pasteur, what was a virus? He didn't really know, in fact. So he was studying rabies infection. He knew it was a transmissible agent, so it was something infectious. But he couldn't culture it in a culture medium in the same way as he had succeeded in cultivating many different bacteria. So for Pasteur, a virus was something infectious, but you can't culture it. So it's a kind of a negative definition. And then really the first useful definition came from Viro1 and I guess another plant biologist who was studying an infection in tobacco plants. And they found that this particular infection, or the infectious material, could be passed through a filter. that had pore sizes that were small enough to retain any types of bacteria. So this was the first clue that viruses were really something different from bacteria. So this was towards the end of the 19th century, beginning of the 20th century, was a definition: viruses are infectious agents that are filterable. So this definition was based on the size of an infectious particle. And what that meant is that the virus particle was less than 250 nanometers in diameter, because bacteria will be retained on a filter with that pore size, but viruses pass through. So that was beginning of the 20th century definition of what is a virus, a filterable infectious agent. Very useful because it gave you a quick test to perform to find out whether you had a virus or a bacterium. However, that's not really a description of virus biology. Okay? So over the next 20, 30, 40 years, biologists began to study viruses, particularly viruses that infect bacteria, bacteriophages, and began to get some information about what exactly viruses are. And Lwoff in 1957 tried to summarize everything that was known about viruses at that time, and it's still what's considered to be, the modern definition of a virus. Okay, so what were Lwoff's criteria? Okay, so he kept the criterion of size of the infectious particle. So for Lwoff, viruses have a diameter less than 250 nanometers. Then he started to look about something that tells us about what viruses are and how they replicate. So the first observation is that they are obligate intracellular parasites. That's their mode of existence. And because they are obligate, I don't know if that's the right way around. So something that's related to their mode of replication is that they don't have the enzymes to generate metabolic energy or to produce their own proteins. So because of that, that's why they are always obligatory intracellular parasites. The host cell has got to provide these two things for viruses. Another thing Lwoff noticed was that, rather interestingly, when you look at infectious particles of viruses, you find that they only contain one type of nucleic acid: RNA, DNA, RNA or DNA, but not both at the same time. That was true in 1957 for all the viruses that were known at that time. Another thing that was known in 1957 is that when you try and study replication of viruses, one thing you notice is that they don't grow and divide like cells. So their mode of replication is very, very different. Now we're going to have a bit of a closer look at each one of these criteria used by Lwoff, because these were proposed in 1957. As we'll see, some of them are not adequate to really separate the biggest viruses from the smallest bacteria. Okay, so we're going to look at a couple of exceptions to some of these rules. Because, of course, things have been discovered since 1957. Oh, what happened? Weird. Okay, so one thing that can be... It doesn't show me. Okay. Oh, it's been... Okay, one other thing we could... it just comes up and then disappears. One other thing that we can add, even if the slide can't add it, is that another interesting difference between viruses and cells is that no viruses code for ribosomal RNA. Okay, so that's why they can't make their own proteins. And this is true even for the biggest viruses. And so some people have even said this is a real fundamental difference in the living world. There's two types of replicators, things that have genomes that transfer genetic information from one generation to the next. There's replicators with ribosomes, so that's cells, right? That's bacteria, archaea, eukaryotes, living organisms. And then there are replicators without ribosomes. These are viruses, and they live by parasitizing cellular life. So according to this view, the history of life is a kind of struggle between replicators with ribosomes and replicators without. And we've been co-evolving with our viruses ever since the beginnings of cellular life. Okay, right, so let's go back to our criteria from Lwoff and have a look at them one by one. Okay, so size of or diameter of the infectious particle. So the very smallest viruses are only about 20 nanometers in diameter, so parvoviruses. There's parvovirus B19 causes a childhood disease, not serious, thankfully, but there are some that infect humans or other mammals. That's the kind of smallest type. A lot of viruses basically have a particle size in between, I don't know, 50 to 200 nanometers diameter. So that's where you're going to find most virus particles, right? It's going to be along here. Parvoviruses, HIV, a lot of them kind of in the 100 nanometers plus or minus a factor of 2. That's where most viruses are. Then at the extremes, you can find some large virus particles like the pox virus. Okay, so the bar here, this is 200 nanometers. So you can see that it's just about less than 250 nanometers in diameter, but maybe 300 nanometers long. And in fact, there are some huge viruses that are visible by light microscopy that infect amoeba. So there are some things that are like half a micron across, and you can see them by light microscopy. Okay, all of these guys, you can't see them in a light microscope. Okay, so basically you can see bacterial cells under the light microscope with a gram stain, but you'll never be able to see viruses because they're just too small for the resolution of the microscope. That's why you need to use electron microscopy to get this kind of image. Okay, so at the high end of viruses, there are some particles that are bigger than this limit that was proposed by Lwoff that have been discovered since then. So the biggest viruses are actually bigger than that. And from time to time, you can see something like this type of object here. So here the scale is 100 nanometers. So it's maybe about 600 nanometers long, but it's only about 150 nanometers in diameter. And in terms of complexity of the structure, it looks pretty simple compared to this pox virus. It doesn't really look more complex. But in fact, this thing is a bacterial cell, a mycoplasma. It's a type of bacterium that doesn't have a cell wall, and its diameter, in fact, is inferior to this limit here. So this can be a problem in cell culture if you sterilize something by filtering. Then mycoplasmas can in fact go through the filter and contaminate your culture, even though they're bacteria. Okay, so the biggest viruses are bigger than this limit of 250 nanometers. The smallest bacterial cells are smaller. Second criterion, obligatory intracellular parasites. Okay, that's true for all viruses. Describes their mode of life. But it's also true for some bacteria. So Mycobacterium leprae, this is a cell that's infected. These are the mycobacteria inside the flagellizosome. Chlamydia, they also infect cells. These reticulate bodies are the kind of bacterial cells that grow and divide inside the infected cell. Elementary bodies are the ones that transmit infection between cells. And rickettsia, and once again, they're intracellular bacteria. This time they move freely in the cytosol. Okay, this is a cytosol of an infected cell. This is the bacterium. So all three of these bacteria cannot be grown in a culture medium. They're entirely dependent on a host cell. So just saying viruses are obligatory intracellular parasites isn't sufficient to distinguish all viruses from all bacteria. Now, kind of together with that, so the other Lwoff criterion, no enzymes for energy metabolism or protein synthesis. So all intracellular bacteria produce their own proteins, and most of them generate their own ATP, their own metabolic energy. Well, except for chlamydia, because in fact they import ATP from the host cell cytosol and export ADP. So they act like a kind of opposite of the of a mitochondrion. So when you look at viruses, okay, no viruses are producing their own metabolic energy. They use ATP and NADH from the host cell. And no viruses produce their own proteins. So they use host cell ribosomes. And the other one that the Lwoff noticed was, well, okay, one of the other ones is one type of nucleic acid. So if you look in a bacterial cell, any kind of bacterial cell, even if it's very, very simple, even if it's an elementary body of chlamydia, even if it's an endospore, which has no metabolic activity, you will always find that the genome is in double-stranded DNA, and that even the simplest kind of cell has got ribosomal RNA, transfer RNA, and messenger RNA. Every cell has got this. When you look at virus particles, well, some viruses have got double-stranded DNA as their genome. Others have single-stranded DNA. Some have double-stranded RNA, and others have single-stranded RNA. So these images here are just electron micrographs of viruses that have got these different types of nucleic acid in their genome. And at least for, they all look pretty similar, okay? So they just look like kind of small round balls of protein. So there's no clear relationship between what the particle looks like and what kind of nucleic acid is on the inside. Now there are exceptions to this rule of André Lwoff that were discovered afterwards. So some large envelope viruses, okay? They might have an envelope, and then a nucleocapsid which has got double-stranded DNA on the inside, and they may have some messenger RNA somewhere else in the particle. So herpes viruses are an exception to this rule. Okay, so this was true in 1957. It's still true for most viruses, but there are some exceptions. Okay, and the last one, which I'll come back to later, is the strange mechanism of replication. Okay, so we'll look at that one in a bit more detail later. Now, before I go on to talk about virus structure and the mechanism of replication, really, how do we know anything about viruses? How do we study viruses? And they're difficult to study, basically, for these two reasons. They're too small to be directly observed, and you can only grow them inside another organism. Now, there's basically two types of approaches that are used studying viruses. One of them is to try and produce virus particles, purify them, and then try and analyze them by physico-chemical techniques to get an idea of what is a virus particle, how does it work, what are different components, and what are their functions. So that's one way of looking at viruses. The other way is to kind of study the way viruses. Okay, so the other way is to say, right, what we want to do is study viruses and their interactions with the host cell. Okay, so in that case, you don't want to separate the virus particle and study it in isolation. You want to study how the virus interacts with the host. So you've got to analyze it in the host cell, in infected cells, or in infected organisms. So in that case, you need tools to specifically detect virus proteins and virus nucleic acids. Okay, so you will use immunochemistry, so antibodies can detect virus proteins specifically, and then different techniques in molecular biology to specifically detect viral nucleic acids. I don't have a slide for this, but just to kind of illustrate the concepts here, okay? So imagine you've got a virus, you've isolated a virus, and you've sequenced its genome. Now it might be linear double-stranded DNA genome, and just from sequence data, you can see you've got, I don't know, 5 proteins that are coded on this DNA molecule. So you might want to find out what these proteins are doing, or at least when they're expressed, okay, inside an infected cell. So you've got the cell here. Here's your virus with its genome. You infect the cell. Then what you do is a kind of time course experiment. Take samples at different times, and you want to look at number of copies of this DNA. And you'll also want to look at the number of copies of each of these RNAs for each of these genes. So you'll have time and then the number of copies of these things. So you might notice that, okay, once you've infected the cell, the number of DNA copies stays stable, and then you get DNA replication. Your number of DNA copies is going to go up. And then you might say, okay, amongst my 5 genes in the genome, I've got 2 that are going to be expressed like this. And that's going to be A and B, for example. And then C, D, and E, they're expressed later. They say, okay, these are my early genes, and these are the late genes in the virus replication cycle. And typically what these things are going to be doing is these ones, A and B, will be involved in DNA replication because this occurs afterwards. And these ones, C, D, and E, will be involved in the structure of the virus particle and its release. These are things that are going to happen later. Okay? So this is the kind of experiment that's been done for all sorts of viruses, and that's how we know about the details of the virus molecular biology. Okay, one of the things you're going to need, though, okay, whichever your experimental setup is, whether you're interested in the structure of virus particles, or whether you're interested in this kind of virus-host interaction, what you need to start with is a purified preparation of viruses, okay? And then you can start to play around with them. So how do we get that? Well, the first thing you need is a production system. somewhere you can grow up the virus. And of course, the first ones were whole organisms. So this was Pasteur in the 19th century managed to propagate the rabies virus from the brain of one infected animal to the next. So in this case, the whole animal was the production system. And it was the same thing for studies of tobacco mosaic virus. Okay, so infected leaves that showed symptoms were ground up and then rubbed into the leaves of the next plant. So the whole plant was the production system. So you get some information from that, but yeah, I tried to change my animation just before in order to bring things up as I was talking about them. And for some crazy reason, they are disappearing as soon as they appear. Okay, so you can, that's got some utility still, producing viruses in whole organisms, especially if you want to study pathology in a whole animal or a whole plant. But you can't really get this kind of very detailed level of information about how viruses replicate. So in order to understand that, you need to get a cell culture system where you can grow your viruses in a more controlled and a simple environment. So of course this was done first for viruses that infect bacteria, for bacteriophages. They were grown first in 1915. For animal viruses, of course, animal cell culture had to be invented before it was possible to study viruses in a cell culture system. So this became possible from the late 1940s onwards with the establishment of the helical line, the first immortalized human cell line. And without that key advance, it wouldn't really be possible to study animal viruses or human viruses. So cell culture had to be invented first before really we could try and work out and understand how human viruses replicated. So that's the culture system, okay? You put the virus in, it's going to grow either in the whole organism or in the cells, infected cells. Then after it's grown up, you harvest the cells that have been infected and make some kind of extract. And what you want to do is purify viruses from that extract. And the classical technique is to use density gradient centrifugation. So the idea here is you're going to start with a kind of mix of soluble proteins, large lumps of cell debris, and from time to time, virus particles. This is all mixed up in the raw cell extract. So you layer this extract on the top of a gradient, sucrose or cesium chloride or other kind of material. So you'll have a density gradient. This is high sucrose in the bottom, very dense, low sucrose at the top. And then you centrifuge this at 100,000, 200,000 G for 16 hours. So the different components will separate out in the gradient according to their sedimentation coefficient and their density. So the soluble proteins, they'll stay up here, or they might just enter the top of the gradient. All of these large bits of debris, nuclei, this kind of stuff, cell organelles, they'll go down to the bottom. And at one point, virus particles will form a band at a particular where the density of the virus particle equilibrates with the density of the material in the gradient. So that's how you can separate out viruses from all these other contaminants from the infected cell. So you get an example here. Okay, so this is a gradient of that's been used to purify pox virus particles. Okay, so this material here, that's where the viruses are. That's the band. So you can harvest this band, look at it by electron microscopy, so you can see that you have pure virus particles. And then you can try and analyze them by a whole bunch of other techniques. So you could separate their proteins and analyze them by STS phage. So that's how you can tell what proteins make up the virus particle. So pox viruses, they're a bit of an unusual example. But it's the best picture I could find of a band in a density gradient. So there are maybe more than 10, several dozens perhaps of different proteins in the particle. For a lot of viruses, you might find one protein in the particle, or 3, or less than 10. Okay, so most viruses, when you do this, you'll probably see less than 10 bands on the gel. A minimum could be 1. The extreme situation is something like this, with a complex particle, several tens of different proteins. But that's how we know, okay? Okay, the other thing you can do, once you've got these purified particles, is look at them by electron microscopy and see what their structure is, okay? And then try and see what kind of structures you get for different viruses. Okay, so virus structure. Now there's 2 basic types of virus particle. Okay, virus particle, or Viro1, means the same thing. Non-enveloped and enveloped. So the non-enveloped or naked viruses, they're composed, they've got nucleic acid on the inside, so you've got the genome, and surrounded by a capsid, which is made out of protein. And that's it. Could be more or less complicated. Now, envelope viruses, they've got the same thing in the middle, nucleic acid, capsid proteins. Often together it's called the nucleocapsid. But outside you'll have a lipid membrane, which is called the envelope. And inside this lipid membrane you'll have viral glycoproteins. Now, if you look at non-enveloped viruses, there are 2 basic types of particles you see in electron microscopy, kind of long filamentous or stick-like structures, which have a helical symmetry. And you have some other particles which are round, which have an icosahedral symmetry. And then some bacteriophages have a kind of mixture of these 2 elements. Now, in amongst envelope viruses, simple envelope viruses, they've got either one of these 2 basic structures, which is wrapped up in a lipid envelope. So this is a rhabdovirus particle. So you can see that inside you've got something a bit like this. You've got a helical nucleocapsid with a membrane around it. This is a cytomegalovirus here. You've got an icosahedral capsid just like this one, but it's got a membrane on the outside. And then, unfortunately, you have some kind of exceptions. Once again, pox viruses, we call them complex. They've got a double membrane structure. And on the inside here, it doesn't really correspond to either one of these 2 simple types of structure. Right, so helical and icosahedral symmetry. What are the basic elements here? So what is the point of the capsid? Well, the basic function of a virus capsid is to protect nucleic acid. Okay? So inside an infected cell, the virus nucleic acid has to be free because it's got to be available to produce direct mRNA synthesis to produce proteins. It's got to be replicated by some kind of enzyme. So it's got to be free inside the infected cell. But outside the infected cell, an RNA molecule or DNA molecule on its own is not going to last very long. In your blood, you've got plenty of DNase 1, you've got plenty of RNAs. Any free nucleic acid is going to be eaten up. So the basic function of a capsid protein is to protect the virus nucleic acid. Okay? So really the philosophy behind the helical capsids is that our nucleic acid is a long linear molecule. Now if I can get a capsid protein that has a tendency to make a circular structure, and I can make circles that go round and round the nucleic acid, and this will form a helix that covers up the nucleic acid, okay, and will protect it from attack by enzymes or just chemical degradation. So this is a very simple way to make a capsid. That's what you find in tobacco mosaic virus. That's the thing that's illustrated here. And it's formed of several thousand copies of just one capsid protein, coat protein. So this capsid protein will form discs. When it interacts with the virus nucleic acid, one of this the double disc will kind of become dislocated, and you get a kind of free face, and other subunits can polymerize. They just add on. And because it's kind of, it's never going to meet up, they just keep on adding on until all the RNA is finished. So that works, very simple. The drawback is that if you want to get a virus that has a more and more complex genome, it means that the particle is going to become longer and longer and longer. And in the end, it becomes unstable. Okay, so this kind of capsid structure places a limit on the genome size that can be encapsulated. Now, the other way of wrapping up nucleic acids is to try and consider, okay, we're not going to consider that the nucleic acid is a line in 2 dimensions. We can wrap this nucleic acid up in 3 dimensions, like a kind of ball of wool or something. Okay, so in this case, the nucleic acid is wrapped up, and we try and package it into a hollow spherical structure. So that's what this is. Okay, the capsid proteins form a hollow sphere, and the nucleic acid goes in the middle. Now, the simplest way for this to occur is if you've got 60 identical subunits making up the hollow sphere. So in this case, 3 copies of the capsid protein make up a triangular face, and 20 triangular faces make an icosahedron. So that's why this is called icosahedral symmetry, right? So if you get bigger and more complex virus capsids, they're going to have more complex architecture, different virus proteins that are in the capsid. But you're always going to have this basic structure where you have some combination of virus proteins that make up a triangular face, and you'll have 20 triangular faces that make up the whole capsid. Okay. Protecting virus nucleic acid, that's the basic function of the capsid. Now, the other function of capsid proteins in a naked virus is to interact with the host cell. Okay, so for a naked virus, these capsid proteins have got to bind to the surface of the host cell. The second function of capsid proteins. However, in envelope viruses, okay, now you've got a lipid membrane that covers up the capsid. So the capsid proteins don't have access to the outside of the particle anymore, right? So that means this interaction between the host cell and the virus particle has got to be mediated by something else. That's going to be a virus glycoprotein. So all enveloped viruses have got to have another element of structure. So they've all got to have enveloped glycoproteins on the outside. Okay? And their function is mediate interaction between the virus, the virion, and the host cell. So that's what the functions of these guys are. Inside you've got the nucleocapsid, that's to protect the nucleic acid. And many viruses have got something in between the nucleocapsid and the envelope. So these can be called matrix or tegument proteins. Often the function of the matrix is just to keep the whole thing together, okay, to ensure that there are physical interactions between the nucleocapsid and the envelope. So it's kind of like some kind of cement that's holding these things together. Okay, that's where we are now. Time for a little break and a short question for you. So I'm going to send this one out on M-clickers. Except I don't have my connection. Great. Okay. Now it's going to be done on a show of hands because I've been disconnected from the server here. Okay, so think about these questions. And then I guess we'll do this one on a show of hands. Okay, everybody thought about these? Yeah, so which of these are true then? Okay, one, all envelope viruses have at least one envelope glycoprotein. Who says yes? It's true. Who says no? It's false. You're right, that's true. At least one. Two, virus capsids are composed of either polysaccharide or polypeptide. Who says yes? Who says no? Most people don't know. Think about it again. Three we'll come back to. Four, main function of the capsid to protect the virus genome. Yeah, that's true. You're right. Okay, I'll come back to five later on. So two. Any more thoughts on this one, everybody? Let's have a revote here. Come on. Just make a decision here, true or false. So who says true? Who says false? People who said false, you're right. Protein. Capsid is always protein. Right? It's easy to get mixed up. Capsule, capsid, right? In a bacteria, you have a capsule sometimes. So a capsule can be polysaccharide or polypeptide. But a capsid in a virus is always protein, right? That's why this question is here, because I know it's easy to make the mistake in terms of vocabulary. So five. Everybody thought about this one yet or not? Okay, who says this is true? Who says it's false? Whoa, interesting. One person who said it's false. Care to explain why? Yeah, okay. It's one. Well, who said it's true? Anybody? Why do you say it's true? Why is it true? Because you had to guess something, right? Actually, this is true. This is generally true. Okay. So for some an envelope virus like influenza, if you wash your hands with soap, that will be enough to inactivate the virus. It's very effective. Okay. But for a naked virus, like a rotavirus, causes gastroenteritis, if you wash your hands with soap, okay, you can get rid of the virus, but it might still be infectious. Okay. Or some might be retained. So why is that true? Okay. Oh, I rubbed out this picture. Think about our virus. It's got to have the glycoprotein in the envelope. This glycoprotein here is going to mediate the infection of the host cell. So if you have a detergent, it's going to destate, it's going to solubilize this whole membrane. So the whole thing is going to come apart, and you won't have any virus glycoproteins associated with the particle anymore. So the capsid in the middle, the nuclear capsid, might be okay, but it won't be infectious because you've taken away the glycoproteins. Whereas if you start off with a naked virus like this, it's the capsid proteins that are responsible for the infection, and it doesn't really care if you use a detergent because it's going to be resistant. Okay, so this is true, right? Last one. 3, viruses are not living organisms. Who says that's true? Who says it's false? They are alive. Some people. Who says neither one nor the other? Some people. Okay. Well, yeah, there's no right answer to this question, of course. They are part of biology, they're part of the living world, but are they living on their own? You can argue it one way or the other, I think. But it's kind of interesting that you can even ask that kind of question, right? So it means that there's a kind of hazy middle ground between cells that are definitely alive and an individual protein is definitely not alive, and viruses are kind of like somewhere in the middle. Probably most people would say viruses are not really alive in the sense that they can never, grow and divide independently. Now, otherwise, if you would say, is a plasmid DNA alive? Because it can replicate, right, inside a host cell. Some of them can be transferred between cells. So if you say a virus is alive, then you've got to say plasmid DNA is alive. And then how far do you go? Okay, we have to move on and leave philosophy a little bit. So now I want to come back to the last element of Lwoff's definition of what exactly is a virus. And because he noticed that when viruses replicate, they don't grow and divide like cells. So what that means is that, okay, you don't have binary division in viruses. So what that means is that for all cellular organisms, if you look at them at some point, you've got one cell, right? It gets bigger, it gets bigger, and then it's going to divide into 2 cells. So at all points in this process, you've at least got one cell that's there. Now, if viruses were to grow like this, what you would have is you would have a virus particle, a virion. It would kind of get bigger, and then these 2 virions would separate, and finally you'd have like two separate virus particles, if viruses were doing the same thing as cells. Now, don't anybody come away from this slide with the wrong idea. This is not true. This doesn't happen, all right? They replicate in a very different way. Now, the first indication that viruses were doing something different came from experiments that were done in the 30s on bacteriophages by Ellis and Delbruck. Now, what they were trying to do was analyze the growth curve for viruses, just like you would do for bacteria. Okay. That is, you start a certain amount of bacteriophages growing in culture. Okay, so first thing, right? Do you want these guys to grow? You've got to give them something to eat. They don't eat culture medium. They eat bacteria. They live on bacteria. So you've got to put them into a bacterial culture. So you inoculate them into their growth medium, which is bacterial culture. Then sample them at different times, and then count the number of infectious phage with a plaque assay. So if you do that, then you get this kind of graph. These came from the original paper. So this is time here, and this is the log of the number of infectious virus particles. So it's for about, 10, 15 minutes, nothing happens. Then they suddenly grow very fast, and then you get a kind of plateau. It's a very typical one-step growth curve that you get for a bacteriophage. And generally speaking, other lytic viruses do the same thing. Okay, so the plaque assay, what is that? How do you count individual viruses? Because we said one of the problems here is that they're too small to be detected directly. Right, how does a plaque assay work? You get an agar plate, and what you put onto the agar plate is a mixture of your viruses and infected cells and, well, non-infected E. coli, okay? You put that onto the plate in soft agar. So what happens is the E. coli, they're going to grow up all over the top of this agar. And if you've got a virus somewhere, it's going to infect one of these cells, a virus, a bacteriophage, it's going to infect one of these cells. This cell will become infected, and then it will release new phage that can infect the bacteria next to it. This is why it's important you've got agar on the top here. If this was just liquid medium, if you did this in a like LB liquid medium, the viruses that are released, they could go anywhere, and you would have the whole all the bacteria will be lysed. But the soft agar will reduce the diffusion of these phage, so they can only infect cells that are really, really close. So you leave that overnight, and what you get is something like this. Okay. This is before you start. These are your agar plates, and then after one night's culture, you see that sometimes you have these holes in the lawn of bacteria. These are plaques, where one virus has landed on some cells. This infected cell has then grown and released more infected viruses, and then it makes a hole in the lawn of bacteria. Okay, here's some more here. There's a plaque. So where your bacteriophage are weakly diluted, okay, everything is lysed. When you've got a lot of dilution, then there are no lysis plaques. And so what you do is you count the number of plaques, you multiply by your dilution factor, and the fact that you've only got 0.1 ml per plate, and this will give you the titer of your infectious bacteriophage in plaque forming units, PFU, per ml. So this is the method used to quantify bacteriophage in one of these cultures. Okay, so just to make sure everybody has understood this, right? Okay. When you see one plaque on one of these plates, what does it mean? Okay. What does it represent? So you've got three options here, okay? And another way to think about this is that if you've got something like this in your infected culture, how many plaques would that produce? Okay. Could be 4, could be 8, could be 5. Okay, so just think about that for a minute, because it's going to be important for you to understand this before we look at the results of the experiment. Everybody got an eye? Everybody decided? Yeah? Okay. So who says number 1? Nobody. Okay. Who says number 2? Nobody. Who says number 3? Some people. Yeah, that's right. Okay. So that means that if this is in your culture, this is somewhere in the mix, right? That you're pouring onto the plate. Just to make sure everybody has got this, okay? So you have a mix of free particles, they're outside in the medium, and you might have an infected cell with a whole bunch of virus particles inside. When this one gets put down on the plate, okay, initially it's going to be one infected cell with a whole bunch of particles inside. When this explodes, when this lyses, it's going to release these phage particles, but they're only going to infect the neighboring cells. So this will give you one plaque on the plate, right? And it's not going to give you 4, right? So one plaque means one free particle, one free phage, or one infected cell. And it doesn't matter how many virus particles are on the inside. Okay, so that's what that means. Oh yeah, I wrote the answer here for you. What an idiot. Okay, so this was the first interpretation of the Edison-Delbruck experiments. Is that okay? You've got a latent phase. Okay, so for a certain time, there's no increase in the number of PFU per ml. Then you have a release phase. This is very, very rapid. This could be like in 5 minutes. You go from 10 to the 6 to 10 to the 7 per ml, or 10 to the 8 per ml. So this very, very rapid logarithmic release. And then it's stable. Okay? There's no more cells to infect. These viruses don't grow outside the cells. So that's why it's stable here. So, I hope this is going to come in. So the first model to explain this was that okay, was it? Okay, we start off at the very beginning. We've got free phage. They're going to they're going to attach on the outside of the cell. They're going to go inside the cell. And then during this phase, the latent phase, they grow inside the cell. So we get more and more. And then during this phase, we have release. So this cell is going to explode and we'll release these infectious phage, infectious particles. That's the first model, right? That's the idea. Now, how can you test this model? Okay, you've got to get access to the infectious particles inside the bacterium. And the way this was done was to exploit the fact that these are E. coli, these are gram-negative. They're very sensitive to detergents or solvents. So if you take this infected cell culture, mix them up with a little bit of chloroform, that will destroy the outer membrane of E. coli, will lyse the cells. But bacteriophage, they are non-enveloped, naked viruses, they're stable, so they remain infectious. So you can lyse infected cells and release the bacteriophage that are inside. So this was what was done in the next experiment. So they lyse bacteria with chloroform, and that means that you're looking at virus particles, but not in the infected bacteria. So if you do that, the dotted line is what we had before. You've got the latent phase, and it goes up. The solid line here is what happens in this experiment when you lyse the infected cells with chloroform. So the release phase is earlier, okay? But what the big surprise is that very quickly, at the early time points after infection, there's nothing. You don't detect any infectious particles inside the infected cells. So that means the first model is wrong, okay? So immediately after the infection, you don't actually have any more viruses left. the virus or the phage itself disappears. You just have an infected cell. And then at later time points, you've got infectious particles that accumulate inside the cell, and then they're released, right? So this is a very, very surprising thing here. So at early time points, the virus doesn't really exist anymore. So this is called the eclipse phase, right? This is when you have early after infection, no infectious particles, no infectious viruses exist anymore. Now it's possible to kind of visualize this process by electron microscopy. So this is from a virus that infects chlorella. It's not a bacteriophage. So the particles are a bit bigger, they're a little bit easier to see. So why does this happen? So that at early stages after infection, so the virus will attach, and then the material that's inside the capsid goes inside the infected cell. So a virus particle that's infectious, it's got nucleic acid and a capsid. Here, they're being separated. That's why you don't have infectious particles anymore, because the nucleic acid is separated from the capsid. So that's what happens at early time points. Then you go into the eclipse phase. You don't actually see virus particles inside the infected cell during the eclipse phase. What you see is an accumulation of later on, okay, you get assembly and then release. So at the assembly phase, you start to see new particles, new virions that are produced inside the cell. And then later on, okay, the cell will lyse and they'll be released. So here you've got some virus. Here you've got virus. In between, you've only got an infected cell during the eclipse. So what is happening during the eclipse phase, okay? So these are the basic elements of the virus replication cycle. This will be true for any virus. It's true for bacteriophages that infect bacteria. True for influenza. It's true for Ebola virus. They've all got to do the same thing. So the first step, attachment. Then entry. Either the whole particle enters the host cell or the nucleic acid. After entry, somehow the nucleic acid has got to be freed up. This is decapsidation. This has got to happen because otherwise this nucleic acid can never be replicated and it can never be expressed to produce virus proteins. So after decapsidation, you get genome replication and gene expression. So this is early steps, then for the first steps in virus replication. Genome replication, gene expression, that's what's happening during the eclipse phase. Then, once you've got a whole bunch of copies of viral nucleic acid and virus proteins, they get assembled inside the host cell and then they are released. So if I was to put this into kind of a human scale, right, it's as if I leave this lecture at 3:30, right? I'm going to go in my car. That's me. I'm the virus nucleic acid, and the car is the capsid. I'm going to go and drive to the Chantier Naval in Saint-Nazaire. So I somehow trick the guardian, right, the security guy at the door to say, yeah, it's me, let me in. I work here. So I enter, right? I get out of my car. I decapsidate, and I go to like the central management place that runs everything in this factory. And I stop it from being a car factory, a shipyard, and I turn it into a car factory. Right? So I get them to make a whole bunch of cars. And I turn all the other people that work there into clones like me. And at the end, we all get into our new cars. We explode we blow up the factory, and then we leave. Okay, that's how virus replication works. Okay, and that's the lytic cycle of virus replication. Okay, so I think that's probably a good place for me to stop. And we'll do bacteriophage and a little bit about human infections on Thursday. So 7 p.m. will be the last microbe of the week.